So I think we're ready to start. I seem to be mic'd as well. So welcome to the next session. This one is on best practices to inform national policy design implementation, perspectives of key stakeholders in the biomedical and public health communities. So this process, I guess, started I guess a couple of years ago now as a debating society, and things are pretty heated. And it's become much more deliberative, and the next step is to actually policy will be made, although not by this group. However, I think what this group says and discusses will have a very large impact, and particularly the NSABB recommendations will play a very large role in influencing what that policy is. So this is, I guess, the time to be quite concrete about what you think would be important to inform that policy, what those policies might be. We're not here to make policy, but on the other hand, we brought together key stakeholders so that that policy can be informed by the views of key stakeholders of communities. And I won't give everybody's bios here because they are in the program, but I think when you read those bios, you'll see that these people are extremely well qualified to comment from their individual perspectives. Ethan Settembre is representing the industry perspective as part of a company that has been heavily involved in pandemic response. Michael Callahan is a physician who treats and organizes the treatment of very high impact infectious diseases in the field and is also very deeply involved in biosecurity matters. Jonathan Moreno is a bioethicist and not a biological scientist, but someone who has a lot to say about these issues. And Robert Fisher is with the FDA, also deals with medical countermeasures, emerging diseases, counterterrorism, and can provide the perspective of a regulator on some of the options for influencing what goes on in laboratories and elsewhere. So I guess we'll go in the order of the program. So Michael, if you'd like to start, we'll have about 10 minutes per speaker, a brief panel discussion, and then open it up to comments from the audience.
Thank you, Phil. I think we're off to a good start because my slides are on a USB drive, which dropped into a boiling hot cup of coffee in Brazil 48 hours ago. And then the second thing that happened is that all of the case studies that we'll be sharing with you today needed to be signed off with something known as host country concurrence or institutional concurrence. The data that we're presenting with you today comes from a number of international partners. And I think what our hope is from our discussion when our group met was to expand the thinking, our thinking here on the NSABB policy activities so we don't have to do it twice or 168 times for each country. That the efforts that are done here are minimally revised in a way that they meet the cultural and scientific mores of where really the burden of gain-of-function activities are really happening. Many of you will consider the truthfulness, if you work internationally and if you work in the industry sector in particular, that the world is flat for bioinnovation. As we know, using our Chinese colleagues as an example, they are sequencing more viral pathogens in 4 months than we have in the entire history of Western Europe and the United States. So the pace and the rapidity of bioevolution and the increased focus on sovereign health security is driving these unexpected types of gain-of-function activities, which are largely beneficent or market-driven. I think we need to keep this in our perspective. So I'm an ex-Fed, and so there's always a lawyer disclaimer slide, and now with an industry responsibility, a Sarbanes-Oxley board of directors slide. But I think the humility that I have with you about gain-of-function and how much we have to do is driven by my experiences with DARPA and its international investments in highly dangerous pathogen management, and to understand what next generation biological threats look like, and to get there early, get there clinically, get there from a laboratory capacity and understand it. But so too does Massachusetts General Hospital also play a role in this, because I compete for federally funded research. I'm a principal investigator for DITRA, Department of State, USAID, and NIH, and run programs internationally through the Center for Global Health. And so I understand what it's like for the academics to be competitive. And for United Therapeutics, therapeutics to make antivirals that will outcompete other antivirals, our drugs need to have flair. So we have a dengue drug in phase 2 that has been acquired by another company. So academic, government, and industry has made me actually uneasy, more uneasy than I was when I worked directly in Gainoffunction many years ago. So this slide we're going to zip right through. It's just to draw your attention to the motivations internationally that spur Gainoffunction research, and it's important to note that they are intentional and largely focused on improving the quality of medicinal products. It should be stated early and quite clearly that the majority of medical countermeasure development is shifting from west to east, and that many of those eastern anti-infectives, most notably the antibacterials followed by the antivirals, are never intended for Western market. So we don't actually see those drugs, and I mentioned this yesterday in the open mic session, but we need to avail ourselves and understand these drugs because we're going to receive those resistant viruses downrange. So we do need to do what I believe it was Andy Weber, the former Assistant Secretary of Biodefense at the Pentagon, stated, we need go-int. We don't need humant, we don't need genant, we don't need mesant, we need to get there, cooperate, collaborate, we need to go. So I think that we need to think about that because you cannot phone this stuff in from Washington, D.C. Our experiences overseas were driven by, and our concern are driven by staffing Asian venture capital. If I go there as a U.S. government representative and I'm in Asian institutions, I see some pretty good stuff. I see non-alarming stuff. I go to venture in Singapore or Taipei, and I see really amazing stuff. So I'm going to show that to you because it will, I think, underscore the importance of penetrating all the different stakeholders that are assisting gain-of-function activities. And the last point is not just the intentional up top, I think that's been discussed, but the unintentional. We need to understand that by not organizing our interventions in a way that's meaningful, culturally acceptable, and meets host country concurrence for sovereign health security of foreign nations, we might have unintended events that lead to problems. And I'll share a couple of those problems with you. One of the big ones, by the way, is that our vaccines are being constructed in a way that guarantees immune escape, immune evasion, and detect or defeat. And there are examples of this that are largely restricted to the poultry sector of Asian vaccines. All right, so I am not qualified to talk about the range of U.S. policy for the NSABB study, but I would just ask that we reflect on the excellent comments yesterday for the non-aligned nations. They are our customers for products as well for public health security. And I think we need to think about those 112 countries, and I'm thankful for the comments that they were brought up because they're the guys that told the Department of Defense Advanced Research Project Agency that they will never bring a vaccine into Indonesia unless it's halal. So we make halal vaccines. DARPA makes halal vaccines. So if you don't understand this at the get-go, you are going to promote incentives for domestic vaccine manufacture with unintended consequences, such as the use of low-cost, whole virus, inactivated vaccines. That is where you grow up the threat agent, the same one we're spending billions of dollars under cooperative threat reduction activities to keep locked away, are being propagated quite aggressively and then killed at the end, and being turned into ways to protect the economic investments of livestock. We're going to spend a couple minutes on this slide because it captures a number of publicly disclosed events which you can read further about, but we also have the references 2 slides from now. The first 2 cases are in your upper left is the Bacillus cereus 1941 and 42 series, and of course Ebola Macano. And these 2 cases are examples of distribution of wild-type agents that have been promoted by a lack of cogent and thoughtful policies by resource-rich biotechnology groups. In the case of Bacillus cereus, most of the pulmonary anthrax cases that I have seen, not cutaneous, pulmonary anthrax cases I have managed, which is 32, belonged to Bacillus cereus, an agent not on the category A list. And importantly, 42% of those patients died. And they died of anthrax because they died of anthrax toxemia in Bacillus cereus. So if you are one of the southern countries that is denied access to the primers to identify Bacillus cereus, G series, anthrax cereus, if you will, you understand why they might propagate the strains throughout their clinical microbiology laboratories so that they can look at the beta hemolytic pattern unique to this anthrax-like Bacillus cereus and is not found in anthrax. It allows them to meet their requirements for host country reporting to the Ministry of Health of 6 Central American nations. So this is a problem inflicted by not sharing reagents to help diagnostic nucleic acid diagnostic capability in very middle income and affluent and medically sophisticated countries. To the center of the slide is Meccano, Ebola Meccano for 2014, where we reflect back that if you collected all of the U.S. Ebola clinicians that have had experience with Ebola Marburg patients, they numbered 103 in 2013. Now there's thousands of them, happily, because they responded to the outbreak. But so too did the chaos of that treatment environment and our inability to do prompt referral lead to clinical diagnosis and the distribution of clinical materials to 8 countries, including Chad, Niger, Mali, and Kaduna, which is the medical center of the Islamic North in Nigeria. It is there Harvard, Hopkins, and Stanford. So the distribution of those biologic samples, which have been kept cold, has just undermined $1.3 billion of cooperative threat reduction activities. My point of these first 2 examples talks about the need to unify U.S. policy between the events that are happening in this room with all the other investments of the U.S. and Europe, okay, is to understand that we need to make these systems work together to provide viable solutions to reduce the wild-type distribution, and that's before we get to bioinnovation. When we think of gain of function, and if you go back to where it was first written, it was talked about in Appendix 3 of the Biological Weapons Convention. Several of you were with us in those rooms in the early 2000s, and I think you will agree that nobody knows gain of function better than a former Soviet biological weapons scientist. They worked on it for offensive uses. Now, our cooperative threat reduction activities are working to drive them from turn their swords into plowshares, and their advantage in entering the Western markets and the markets of the SFU are to be better than we are, and to test those medical countermeasures against the most robust and hardened threat agents, particularly in the filovirus cases. As an example, us in 2002, back then, we could run around viral hemorrhagic weapons labs at Vector. So we could do that then. We could work quite closely with our partners then, and right now, due to policy issues and politics, we're unable to engage them in really meaningful discussions like this one here. The best case I have for you comes from BioVenture, and it is located at the 3 o'clock position here. I'm sorry, the laser pointer is a little bit meek. And that's a product that we found out about through Bioindustry and venture capital in an unnamed city in Southeast Asia. It is not China, it is not Taiwan, it is not Japan, but it's one of the most affluent. And they were fielding market entry, radically destabilizing technology advancements that would take over large markets. And the example that they used, driven by a McKinsey consultation service and a bunch of Wharton school graduates, MBAs, was in order to go after the very lucrative cosmetic industry, the Botox industry, you needed to go after the #1 thing that the patients hated, which is repetitive shots. And you do this by altering the botulinum toxin to make it last longer. So it did not show up in the PowerPoint slide is the SNAP25 cleavage site of the alpha subunit of the botulinum toxin, which is located about halfway down the stem. And by hiding it from serine proteases in your body, it basically produced 3x increase in the physiologic half-life unbinding. Okay, so it was not nefarious. It was economically driven, practically by business reasons, and poses, obviously, huge gain-of-function examples to us. They appreciated this group of young scientists, appreciated they did not need to make it more virulent. So they wisely came to the cultural understanding that actually Roseanne brought up from McGill earlier today, that there's a cultural issue can really help you here, but they missed on the other part, which is longevity. Two fast ones just to end is that we also need to understand the self-infliction of not providing viable alternatives in the very rowdy avian poultry vaccine markets of Southeast Asia, where the vaccines are created locally. And as new clades of low path and high path H5 come through, and basically Newcastle's and avian botulism and avian cholera, which decimate the backyard terrestrial protein herds, and that these are critically important to the maintenance of protein security in the Islamic Republic of Indonesia and Malaysia, we cannot deny them the opportunity to come up with reasonable low-cost vaccines that are generated at pennies per dose, rather than the international option, which costs $7 a dose per bird. That chicken in Indonesia has a half-life of about 6 months, and no one's going to invest in that foreign import, so they'll create the vaccines locally. Rather magnificently, they are pretty good at recombinant technology through segmental reassortment. So they take their H5s, both the new one found in Aceh and the current one found in Bandung, and they do in vitro recombination and get reassortment, and they select for it. And only after they get a winner do they go through any sequencing technology, which is perfunctory and not the purpose of the conversation. My point here is that if you want to prevent recombination to make, which in this case is producing a vaccine that will last longer through subsequent rounds of future avian attack on the poultry, we need to get in the game and provide reasonable alternatives. Department of State did that with the purchasing and co-licensing of the H5N2 vaccine, which allows us to look at H5N2 vaccinated birds, and if they prove that they're efficacious to the Indonesian government, if they develop H5N1 antibodies, which could only have been conferred if they saw the wild-type pathogen and the chicken did not die. So vaccine strategy, it's called DISMA, which is internationally renowned by the FAO, allows us to provide a reasonable technology that will prove metrics inside these regional economies. The last point is just, how much taxpayer dollars we wasted trying to take highly dangerous pathogens and locking them away in freezers in Africa, and then not understanding that if you want to protect the livestock herds of the Nigerian north, all those vaccines are grown anthrax that is turned into dead product that is injected into those sheep. And this is an example of sheep red blood cell agar quantification of colony forming units from just a couple years ago. Let me just end and bring this together with just some policy alignment that we might act on, which might crystallize what we're doing here in the acknowledging West that will scale perhaps a little bit more reasonably to the emerging economies and to our collegial bioventure communities on the ASEAN. The first thing is we have to engage bioventure. If you look at all the cool stuff, and that's a euphemism for things that scare you, the cool stuff is funded by venture. It is not typically Ministry of Health funded or regional NGO funded. It is funded by venture. There's a large number of this. They've been produced, of these cases, they've been produced and distributed through certain agencies of the US government, but they need to be put before the academic experts and industry experts. They need to read these things to understand their implication. Vaccines, we need to provide licensed, safe, and effective and inexpensive vaccines that don't require cold chain. All vaccines made in Indonesia and Malaysia for the agricultural market are cold intolerant. So what they need to do is, this is one way in, is to thermal stabilize vaccines, technologies that we can bring to them. And why not just give them export vaccine design? In an example that is 4 years old now, the government of Indonesia allowed the sequence, not the virus, to leave Indonesia, come to a US federal agency. Just the sequence for hemagglutinin was turned into a subunit vaccine product, which was GMP, went back to Indonesia, and was tested on birds that were challenged by wild exposure, okay? So we live in an age where sequence in place is becoming the norm, where pathogen exportation is definitely looked down upon by 112 nations belonging to the non-aligned nations. And so we are going to see fewer of these virulent viruses being sent to our major facilities of Europe, United States, and Canada. And so if we understand that, we can roll with it. We can adapt and provide bioinformatics, big data analysis system, and distributed manufacturing based in the synthetic and cloud space. The last 2 points to end with is just the U.S. government performers. We spend a lot of time at USAID, DARPA, Prophecy, and Center for Global Health protecting our international collaborators from R1-funded investigators who seek to do nothing more than get a virus, go home, and write their big Nature paper. And if we do not start putting into the source selection criteria of awarding those contracts into building capacity and maintaining stable, not unstable relationships with our international partners, then we're going to perpetuate the problems that we're having right now with Ebola, the problems that we're having right now in Zika. Do you know how many strains of Zika we have in the United States? 9. Do you know how many Brazil has deep sequenced right now? 61. Do you know how many Panama has right now? 37. Why are they averse to sharing them with the United States as members of the UN nation? The answer is they've been ripped off. Okay, they've been ripped off, and they will tell you these cases, and they'll point out who the offenders are. So we need to think about that as part of the NSABB policy for our federally funded researchers. And the last is just incentivizing our host nation compliance with the policies that are happening in this room by doing what we failed to do for all of our CTR years in Russia and China and Brazil and South Africa and Nigeria, and that's provide a metric for these activities that they measure that has value at home. It saves chickens, it saves the cattle and livestock, it reduces children, infant mortality, it protects mothers that are pregnant, okay? And so I'll just end with further reading. That's your reference slide. This is all no longer for official use only. These are publicly available documents, mostly on the website, and I'll turn your attention to Sequence in Place, the engagement strategy for maintaining sovereign health security, which is one of the premier documents that is missing from our dialogue here. I went a minute over, I apologize, and I'll wait for your questions at the end of the session. Thank you so much.
Thanks, Michael. Robert, if I think you're right.
So first, I'd like to thank the invitation for FDA to come up and speak with you and to participate in this panel. I like to think that FDA is somewhat uniquely positioned for this discussion. In part because we do research, we do fund research. Believe it or not, we actually have laboratories, we have capable laboratories, we have talented scientists, but the real reason I think we're here is because of our main mission, which is to really ensure that the medical products and all the other things FDA regulates that are made available to the American public are safe and effective. And we have a variety of regulations that apply to how we evaluate these and how we make these available through our regulatory mechanisms. This can range from the randomized clinical trial, where you're evaluating it in patients, to the use of surrogate endpoints for accelerated approval, and if there's no other way to do it, there's no ethical or feasible way to use those other mechanisms, we can actually rely on animal efficacy data in some circumstances. Regardless of the regulatory pathway that's being used to push a medical product forward, the FDA needs to rely on data. We need evidence. We can't determine if a product is going to be safe and effective without the appropriate science being done to provide that evidence. So with that said, if the gain-of-function framework that's under discussion is really framed in terms of looking at agents of concern or particular pathways of concern, the vast majority of things that come into the FDA aren't going to be impacted by this. Now, with that said, that very small part that does are potentially high impact, right? Which would be things like pandemic influenza, coronaviruses, and those unknown threats. I think I can provide a couple of examples of circumstances under which there may be some impact on the FDA from the framework. And I think actually Michael did a really great job of setting me up for this, because some of these tie right into what he was just saying. The first is for the production of vaccines, a lot of times the seeds are produced from molecular clones. Okay, and depending on how those molecular clones are generated, I don't think it's hard to imagine that that could actually fall under the gain-of-function umbrella, which then has implications in terms of how quickly that can happen. Also, if you consider that, and we've seen this before, if you have a circulating strain of influenza, pandemic influenza, and you take that and you try to grow it in eggs or you try to grow it in cell culture and it doesn't want to grow, well, now you're in a situation where you're going to have to adapt that virus to grow in the cells, the eggs, that are used for the mass production techniques that will allow availability of large amounts of vaccine that can then be used to protect the public from that threat. Now, how do you get around these challenges, right? How do you keep things from getting caught up in the framework and getting delayed? Because in a public health emergency, you don't want that. I mean, we've seen that with influenza, we've seen that with Ebola. Well, at the October 2014 NSAB meeting, someone actually said that potential high-threat gain-of-function research should not be pursued without access to vaccines or drugs that can abrogate or ameliorate disease. I think another way of thinking about this would be to consider that risk associated with high-threat gain-of-function research that's deemed to be critical to understanding a pathogen or deemed to be critical to making these countermeasures available in a timely fashion. This can be mitigated using the appropriate stringent safety controls. So with that, another tool that the FDA can bring to bear when we're approving drugs, vaccines, medical products, we provide advice to our sponsors, either through frequent meetings or through our published guidances. And we also meet with our USG stakeholders to make sure everybody's on the same page and cooperating in terms of the public health emergency countermeasures enterprise. It's always better to deal with these challenges early in the medical product development pathway than it is late. We don't want something to come across the FDA's bow at the 11th hour. The advisory, having a vaccine advisory committee meeting probably isn't where you want to start having these discussions. I think, the FDA is looking forward to continuing to work with the relevant stakeholders and our USG partners to make sure that and to support a framework that will allow the flexibility so that when we are faced with these public health challenges, we can respond appropriately.
Thank you very much. Thank you, Jonathan. Yeah.
So when I was invited to be on this panel, I do what I almost always do when somebody from the academies calls me to ask me to do something in this building. I said, sure. Then I realized that I was going to be on a panel with people who actually knew what they were talking about, and that raised some concerns. And then I saw that I was on a stakeholder panel, and I, of course, asked myself, well, what kind of stakeholder am I? What's my stake? Now, I guess the obvious one is, well, the bioethics professor, but I've decided to be a different sort of stakeholder today, trying desperately to lower your expectations. I'm a healthcare consumer, somebody who used to be called a patient, and therefore I'm very much at the mercy of people like most of the people in this room who do know something about these topics. And I tried to find a slide to succeed this one that I would have labeled one of the great unwashed, but I couldn't find a really polite one. But that's what I am. I first heard about gain-of-function, I think not long after the term was coined, which I believe was around 2011 or 2012. And I realized that if I was going to talk about this in future, I would have to learn something. So I'm putting myself today in the role of somebody who's not a scientist. I'm a philosopher and historian, and try to summarize some of the points that I think I have learned about in the last few years, and I think I have seen a developing set of conclusions, rough conclusions, I hate to use the word consensus, that largely the community that I've observed, namely you, have reached, and some outstanding questions. So let me try that. Along the lines of lowering your expectations, there's this concept in the philosophy of science called fiduciary knowledge, and basically what that means is we all rely on other people who really know something. We rely on their judgment and their ability to work together and figure out, as one particular presidential candidate has said, if we all work together, it's going to be beautiful. So I'm relying I rely on you, just as we all rely on the architects and the engineers and the mathematicians, right, who put this building together, that this exquisite room will not collapse while we're sitting here talking politely with each other. So I'm in this position, you know, I'm trusting that, for the most part, that the experts will actually get to the right place. So let me summarize quickly what I see as some of the outstanding questions for you that you have raised, and that one hopes that the report ultimately in SABB and whatever the academies come up with will help us. So gain of function, not a great term. I think there's pretty much good agreement about that. It doesn't really hit the mark. It reminds me a little bit of the term cloning. Now, gain of function hasn't gotten on the cover of Time magazine. George Lucas hasn't made a movie about gain of function yet, although just wait. Nonetheless, like cloning, you know, nobody really liked it. And certainly the people who met in this building 10 years ago to try to sort these things out didn't like it particularly. But it got a certain purchase in the public discourse, and gain of function has kind of done that too, but it doesn't seem so great. I'm really happy to hear that Botox, thank you, Michael, we got gain of function from Botox. That'll save me some money over the next few years. But it's not a great term. There are always gains and losses of function. How do we can think about natural mutations in this context, escape mutants that are resistant to medication? Since the term gain of function is fundamentally not the best term, it's very hard to arrange policies. You know, the intuitions don't flow well, so you have these policy issues. Lots of talk, of course, I've heard over the years about safety records and lab accident risks, and how much do we really know? My postdoc, Nick Evans, has written about this. So how good is the reporting? Probably not so good. Between the lines also, people worry about what biological safety level ought to be required, some sort of BSL-3 plus or BSL-4 minus. These seem to be outstanding questions that could be addressed. Lots of disagreement about are there acceptable alternatives for vaccine development? Are gain of function methods always necessary? And of course, there's the time lag between a pandemic like Zika or Ebola and how do we need basic science as compared to ring vaccination and some of the things that or isolating affected communities. Nonetheless, there is some agreement. Much regulation fails to hit the mark. I think I've mentioned that already. There was the Novartis example a few years ago. But everybody agrees, I think, that some regulation is needed. Even I am subject to regulation. I do survey research once in a while on informed consent in clinical trials, or I have done that, and every couple of years Penn reminds me that I haven't taken the quiz to get recertified to be an investigator at Penn, and I get very annoyed, and then I have this pogo experience, you know. I am my own enemy. But I generally take that quiz, and sometimes I pass. Biocontainment, not great. Can we do better? Not a perfect record. Everybody agrees that there's some stuff that gets out that ought not to. And then there are the human problems, which I'm really impressed by the remarks this morning about the fact that so many of the errors are human errors. The same is true in cybersecurity, by the way. Most of the problems we have in cybersecurity seem to come from human beings, not from the systems themselves, which I think is interesting. People have said over the years that really it's a pain in the butt, but you can do risk mitigation. You have to think about it a little bit. If you really want to do some work, there probably is a way to make it a little safer. Just have to think about that. And sometimes there are alternatives. They may not be the best option. We may not be able to answer all the questions in science we'd like to answer. That is not a new fact in the culture of science, but maybe we can come up with alternatives that are nearly as good. The looseness between genotyping and phenotyping, I think that's very interesting. I generally get vaccinated, I guess, for flu every year, but this made me realize why I sometimes don't bother, because I'm not so sure. Is pre-pandemic strain selection transformative? This is a big discussion a couple of years ago in this room in new vaccine development. I think the consensus seems to be, well, it's not short-term, maybe not even mid-term, but in the long term, we need to leave a space for something like gain-of-function for long-term improvements in vaccine development. Human beings are vulnerable right now. I've heard several people give examples, for example, about SARS-like coronavirus. That's a problem right now. Maybe there's some areas in which we should be moving faster and other areas in which we should be moving a little more cautiously. And then animal model development is another example I've heard people talk about. So, trying to say something constructive and relatively concrete. I was thinking of what it would be a new way to do real-time assessment in basic science labs. So what would be the triggering criteria? NSABB has come up with these 3 criteria. I recognize that there is a debate about what the logical connective is. Is it an and or an or? It seems to me, speaking of public engagement, this is a fantastic opportunity to do targeted public engagement. Would people like me, members of the Great Unwashed, would they tolerate the condition that all of these criteria should be existent at the same time, or would they really prefer us to be more conservative about this and only one standard, one criterion at a time? I think that's a great opportunity for some kind of public deliberation. So what would be the precedents for the model? I'm building up to something I want to suggest. Those of us who've done work in venture capital as consultants, or when I did a project with Gates a few years ago on their global health work, I was struck that they had adopted a milestone. So perhaps we could ask our scientists who are doing a project, what's a milestone in this project that you think you yourself would like to see a moment of reflection in whether you take the next step? What are the decision points, as it were, for you to decide whether you go further in this project? Again, this is all based on the notion of the triggering criteria that I've had up before. We see this in phased clinical trials that IRBs are supposed to oversee. Innovative surgery, generally it doesn't happen. It's not supposed to happen, in my view at least, with many candidates at a time. You do one patient at a time and reassess. You do 2 or 3 and reassess. And then, of course, the DSMB model is based on the notion that there are limits on continuing. So I'd ask in this new model, I would ask the investigators to set up some milestones. I was very interested, there was a brief discussion a couple of years ago about this particular practice, the Biosecurity Task Force at the University of Wisconsin, multidisciplinary team, basically focusing on the higher risk or big magnitude labs, and establishing and applying risk mitigation principles that are communicated, discussed with the team. So there's another model as well. I'm going to give a plug to our colleagues at Stanford in bioethics, this benchside ethics consultation service, some of you know about it, where a geneticist, ethicist, law professor will actually be on call for the team in a lab, and they will go and talk to the PI and her or his team, and they'll talk about what they're doing in the lab and whether this is a moment for further reflection. That seems to me to be that, along with the previous model in Wisconsin, they've caused me to think about what I call RBATs. Now, RBATs are going to drive people bats. I didn't think of that, at first when I made up the acronym, but it does work pretty well, and I think it's probably true. Nonetheless, this is aspirational. Maybe for various reasons it can't be done. I heard a number of people over the years talk about the need for some kind of dynamic, real-time, iterative process. This is for the lab. This is not going to be for the application process if there is a pandemic. Perhaps based on milestones that the team itself would develop. maybe unannounced audits, nobody likes that, but maybe yes, maybe no, depends on the situation. There would be case-specific risk-benefit parameters. A lot of people have said in the last few years we really need to go do this case by case. Maybe this may have some educational function. It might be voluntary, it might be mandatory, depending on what the funder wants. That I thought that some idea like this, to actually bring people into a lab, something like what goes in Wisconsin, something like a hybrid of what goes on at Stanford, might be something useful for the lab. Now, of course, I don't have in mind that these are going to be dispositive. They're not DSMBs or IRBs. The notion would be, and I've done this when I was on staff of advisory committees, presidential advisory committees, when you're working with agencies, you ask the agency team to tell you, how have you proved to us that you have considered these questions? So what are you doing? What are the questions that occur to you? What are you doing to address them? That's the spirit of it. It's a collegial, iterative process. And this would be more than paper compliance, would be a dynamic, real-time process. The bodies actually go in the lab and talk to people. Problems, pretty obvious. More bureaucracy, you might say, although in my ideal world, I think of this as colleagues going in to talk to other colleagues. Obviously, there's payment issues. The Stanford group got some funding to get off the ground for this, and I think they have some ongoing support, external support. And it doesn't solve the rapid response problem for the emerging pandemic virus. Not intended to do that. But it's intended to address some of the concerns that people have about the utter independence and non-transparency of what's going on in a lab that's doing work that meets one of those criteria. That's the news from the Great Unwashed, and I look forward to hearing more of the discussion. Thanks.
Ethan?
Yeah. Thank you.
First, I'd like to start off. I'd like to thank the organizers for inviting me to speak on this topic. I am indeed the Ethan Settembre. I'm the head of research for Securus. Securus is a flu vaccine manufacturer, which is a combination of Novartis vaccines and also BioCSL. And I point that out, obviously, to say where I fit as a stakeholder, but also I'm really part of what is a large global system to generate flu vaccines in a timely manner. And I'll describe the system as a whole to put it in context, and then, of course, the role that manufacturers play, and then some of the gaps that exist where improvements can occur. And those sorts of improvements, I'll describe them in some degree of detail. So this is just a sort of brief snapshot of the global influenza system, and indeed it's a system that involves 141 national influenza centers from around the world where samples are obtained. They're sent, perhaps sequenced there, but generally sent to 6 WHO collaborating centers, which are also distributed around the world, where antigenic characterization takes place, as well as sequencing to determine whether something's new, so we've seen it before, whether or not we have existing vaccines for it. And indeed, there's a number of other processes that happen in those centers and other related centers where there's various events to make a vaccine virus strain or a candidate vaccine virus, all before it really gets to the manufacturing site. And then the manufacturers, of course, generate the vaccines using a variety of methods, generally eggs or cells where the virus is grown, inactivated, and then perhaps further processing. And then finally, there's still a release that occurs both within the manufacturing, but through a series of essential regulatory labs that also themselves have to make a series of reagents which are used and distributed around the world. And at the very end, of course, there's a vaccine, but that vaccine, in that case, would just be sitting in a manufacturing site. There's a whole series of steps that then occurs where it needs to get to people and actually get to be used, because after all, the best vaccine is the one that's used. So you can see this is quite a complicated system with many players, both public and private players, which leads to, at a variety of steps, propagation delays and other aspects just because of the nature of the system itself. And so that is how the system is set up. And then one may ask, well, what happened most recently in a pandemic situation? So here we have a prime example relatively recently of the 2009 H1N1 pandemic. And actually in this effort, the global system worked quite quickly, considering the virus itself was first recognized March 18th in Mexico. And actually it was by June 11th that there was actually the declaration of the pandemic. But before that, there were, in late April, clinical virological assessments, then vaccine strain preparation, testing, evaluation, initial seed viruses. Some of the seed viruses involved reassorting, which is a process by which the HA and NA generally, although it could be more than that, but the HA and NA of the candidate, of the virus of interest, is reassorted with a known low pathogenicity common backbone, generally PR8. And ultimately, those were transferred to manufacturers. And you can see in each step, there's many hands that are involved. There's places where handoffs occur, and there's a lot of work that needs to be done at a variety of groups in concert, which is very complicated. And you can see that it wasn't until although clinical trials actually were running along, it wasn't until the end of the process of batch release, which finally occurred in September, and then the shipment of the first doses out to where they needed to get to people. And of course, that was the ultimate goal. And this was quite quick in the overall scheme of things, 4 to 5 months, particularly in the case of a pandemic. And but what does that look like? Even though the system as a whole worked quite well in moving forward, getting a pandemic vaccine out, if you take a look here, what you'll see in the black is an increase based on the number of cases per week in the United States. And you can see that represents the peak, the wave of the H1N1 pandemic sweeping across the United States. And what you'll see, that yellow bar that begins to increase at around September very slightly, and then really more meaningfully into October and further, that's the provision of the number of doses that were delivered. And so what this sort of represents, and I think it's pretty clear from where you're sitting and certainly from where I'm standing, which is that we were in the situation of vaccinating the survivors of the first wave, because actually no meaningful quantities were really available prior to the peak itself. And so there's a real need for speeding up, particularly in the case of a pandemic, although used in seasonal cases as a seasonal situation as well, to speed up the production. And there's a variety of ways to do that. And in fact, it was because of this that at the Novartis vaccines in particular, we took an interest in determining, can we speed up this process? And the process, the place where we noticed that great speed could be added would be at the front end of the process. And initially, the start for a manufacturer of the process of generating a vaccine is waiting by the mailbox to receive the vaccine virus from the collaborating centers or from the center that you'll receive it from. That's one way to start. An alternative is, with the availability of HA and NA sequences, is to generate the vaccine virus itself on an appropriate backbone that actually could work well in your process. And so at Novartis Vaccines, we worked with the J. Craig Venter Institute and SGVI to come up with a synthetic process for generating synthetic influenza viruses, where it's designed that they are actually on the backbones which would be attenuated, but would therefore allow for speed, accuracy, and ultimately they'd be designed for high yield. In all cases, it would be they would be attenuated. The backbone itself is an attenuated backbone. And so it's one of the ways that we consider that we're actually generating vaccine viruses that can be used and really address an immediate, important medical need in a short period of time. So something where it's really essential that we deliver ahead of that wave. Now, this was work all that was done following the 2009 pandemic, but we had a more recent opportunity, and this was in particular for H7N9, where we could apply this actual methodology to determine how quickly it can work in a situation that would be similar to what would be in a pandemic situation. And in fact, using that methodology, it was actually March 31st, Easter, where the viral coding sequences were first posted online on GISID by the China CDC. And this information sharing is really what can enable the beginning of the process of being able to move very quickly into the process and create proper countermeasures in a reasonable period of time to address the needs. And you can see that working with our colleagues, actually, who were in California at the time, based in Cambridge, they began synthesizing the genes. Their HA and NA gene sequence started moving into April 2nd, where gene assembly was complete, and it was shipped, actually still in the mail to us, where we've generated the viruses themselves and saw first rescue on the 6th of April. So you can see that's a big time difference, especially considering that it wasn't until April 11th that the CDC received the wild-type virus, which would be the start of the process for generating the conventional vaccine reassortants. And so this is just an example of the time that one can cut off from the traditional methods by which one can deliver a vaccine so that it's used in an immediate need situation, not just for a pandemic, but actually more broadly in seasonal situations as well. So I would argue that from the perspective of the way in which the work is done, that this is one of those activities that would not be considered gain of function of concern as it relates to particularly the concerns of getting it to people in time for use prior to a pandemic wave. So I leave you with that, and thank you very much. Happy to take questions along with the panel.
Thank you, Ethan, and the rest of the panelists. I want to make sure we have plenty of time for public discussion. So I think we won't go through all these questions, but I think maybe 3 of the questions quickly, and we don't need to go down the whole line. This can be more like a quicker meeting if you feel inspired by a question and have something to say, that'll be fine. The first one is, there is some consensus that there seems to be that not all gain-of-function research is gain-of-function research of concern, and not all of it requires new policy. This does not seem to be the case where you know it when you see it, because it's clearly that different people can look at the same experiment, and some would say it's of concern, and others not. Are there any thoughts from the panel on what does constitute an experiment of concern in a way that's sort of implementable, that you could actually define in some way of what the proper scope is?
Okay.
Thank God.
I was going to draw out that pause. So much power there for a minute. Ethan and I had a little quip to the side here. He might not agree with this because he didn't have a chance to respond, but we note at the beginning that, we know that the actual trigger mechanisms for what is gain of function are actually one of the major motivations to push gain of function, and that's that there is no medical therapeutic. Mm-hmm. And so that's the criteria here, but it's exactly the driving force that that's a new unmet need market. So there's a twist when we try to explain this internationally, that we don't want you doing gain of function research because you don't have a medical therapeutic. And as the Russians said to us, that's the point. So from a pragmatic standpoint, we shouldn't have to be like them to understand the optics externally. We should critically review it for its complete end-to-end reasonableness. So as for what I consider, gain of function, is at the title we're going to be meeting again and again because new technologies are changing what qualifies as gain of function. Remember that we're the country that invented, the U.S. perceives it's the country that invented artificial amino acid encoding, but the only groups that have turned that into products are 3 Asian countries. Okay, so we're way behind. And so gain of function in the synthetic biology space, if we use that, is we don't even have the gene code that allows you to organize artificial amino acids to confer novel folding patterns in proteins that can do novel things that were never allowed in nature before. So this is referred to as molecular hysteresis for the virologists in the room, and is a big concern, and we barely have that technology in the United States. That is an industry proprietary technology to produce winning, enduring, highly resistant proteins. So I slipped a little bit away from answering the question by telling you we still won't know what it is next year because there'll be a new technology that we will need to worry about, that we need to contend with. So our definition needs to be general, adaptive, and culturally appropriate through foreign scientific communities. Did you agree with any of that? Sorry, because it did say.
Yeah, no, I would agree with some of that. But, you know, I think actually it came up in more of the discussion yesterday, somewhat around risk and benefits, and the difficulty in defining really what the benefit is. There's the difficulty, obviously, in the risk as well, but the difficulty in defining the benefit. So I think that really it has to be a bit of that discussion around, and perhaps that relates to, you know, we need to do this work so that we can generate the countermeasure, and so it's a little bit of back and forth. So in either case, I sort of link those two together, the risk and the benefit, which is very difficult to determine.
Yeah, I think we really need to look at it from what's the consequence, right? Because if you're looking at the consequence, that can be informed by what's the consequence of doing the experiment and dealing with that result versus the consequence of not doing it and not having a countermeasure when something happens.
I was thinking, if I'm a member of Congress, I would really want to be sure that you know how to keep this stuff in the lab until you come up with something that'll fix it. So the containment issue, I would say, as a member of the public, is, you know, we've seen this before with recombinant DNA. That was a big thing in the 70s, of keeping it in the lab, and I think that's still true from the point of view of the general public.
Yeah, I remember when I was showing my age, when I was a kid, I had a copy of World Book. Remember, it's like actual encyclopedias, and one of those editions, the science year, actually was discussing recombinant DNA research, and what they were talking about for recombinant DNA research looks a lot like what we have for our BSL 3 and 4 labs now.
Maybe this is related to the next question, and given that there is still definitional uncertainty over what is gain-of-function research of concern, among the tools that are available to influence what goes on in the U.S. or outside the U.S., U.S. government funded or non-U.S. government funded, what do you recommend for tools? And the RBAT idea is, I think very interesting, but are there other tools that you would want to introduce, or some that might not be appropriate given this situation?
I'm only good for one tool a day, so tomorrow it's on everybody else.
So many of the tools are so encumbered by proprietary intellectual property that they're intentionally not being made available. And I use the reagents that are used in our own industrial processes as well as those of our collaborators overseas. They have competitive advantage from having an unfair advantage by having a critical reagent, access to a strain to make a hemagglutinin that confers immunity across many clades of H5. And so, we have to change the incentive plan to produce more players, more stakeholders, and therefore more solutions. So I think we should, again, listen very carefully to industry, foreign industry, working with foreign markets. And I hasten to add that a growing number of this innovation is for therapeutics that are never intended for USDA and FDA approval. So they just aren't. The market is elsewhere.
So kind of taking off my FDA hat a little bit and speaking as a scientist who's dealt with select agent regulations, who's dealt with DIRC, who's dealt with some of these things, from my own perspective. One of the things I would like to see, and I believe you touched on this, Jonathan, was to bring all these frameworks together so that we're not talking about gain of function in a vacuum, away from DIRC, away from all these other things. And I believe yesterday one of our European colleagues mentioned how the EC approaches this, and it seems they have a more global, unified approach to looking at the big picture in terms of, this is your research, what's the impact?
So that brings us to the last question before opening up the mics. and that is, I think we're reasonably clear on the intended consequence of the new policies, and that is to protect the public from the unintended consequences of gain-of-function research when done for beneficent or economic purposes, or the intentional consequences if done for nefarious purposes. But as new policy is being considered, in addition to considering the intended consequences, it's important to consider the unintended consequence of new policy. And are there any unintended consequences that you might foresee or particularly be concerned about that you would want those making the policies to be aware of?
Well, certainly for me, from the global system perspective in influenza, many of the processes that are commonly performed really, depending on how one reads it, could be considered as gain of function when really they shouldn't, because they don't really fit the categories. And in fact, if you weren't able to do the traditional reassorting that's done, generally we would not be able to have enough vaccine for those who need it in a timely manner. So for me, that would be one of those unintended consequences.
I'm fine with that.
I think, oh no, please.
I was going to say, sometimes I think it's better to pay to piper now versus later. For example, if you're talking about a gain-of-function experiment that could inform rational vaccine design, if you do that now, that may preclude the necessity to do those gain-of-function adaptive experiments later. So in terms of the unintended consequences, I think anything that results in uncertainty or results in knowledge gaps, I think those need to be carefully considered.
Thank you. I think maybe with that we'll open up for the mics for questions from the audience.
Hi, Wendy Hall, Homeland Security. I had a question, Mike, for you. I appreciated your comments yesterday reminding us of China and the innovation and the values that they hold might not be equivalent with values of the universal scientific community or the U.S. And I'm looking at being a recipient of the NSABB and other recommendations that come in from the public to shape policy. In that discussion, what is the best way to make sure we're getting the full picture of risk and benefit, pros and cons, and importance for governance, if to include the private sector dimension and the venture capital dimension? Because as you know, the USG is tapping into the NSABB for input, mostly from the academic community. I don't know if we have an equivalent tool in our policy toolkit to get stakeholder input looking at private sector equities. Any ideas?
Great. And hi, Wendy. So nice to see you. I think we heard a little bit about the instruments being used to manage gain of function throughout the ASEAN, and George Gao made the important point yesterday if they're actually policing their own systems with nationwide recommendations. And the case he used was to prevent viral twinning studies, to prevent segmental reassortment. So that was nice because it talked about enforcement. We haven't talked about enforcement here really, but in terms of the ways to more thoroughly engage them, for your current position, for example, is you have to understand all the stakeholders and their motivations. We have our successes internationally in reducing gain-of-function studies where these accidents occurred because they're driven by other issues. For example, for the DoD programs, DARPA programs for the Blue Angel, which was the pandemic H1N1 rapid vaccine system, is that vaccine needed to be made halal or we were not going to transition it into an Asian-based manufacturer. So that was an early engagement, and having the Islamic clerics tell us all of this and tell us what's halal and not halal, making sure the expression system didn't make nicotine. It's nicotinia, by the way. So it was very important, but then we got it exactly right. And because we got it right, we shut down a major investment through the ASEAN in halal vaccine manufacturing. We didn't know we did that. We undercut them when we were earlier because of the speed of this platform. So accidental, but we'll take it. We'll take it as a win. I was quite emphatic about the role of Asian venture capital and FSU venture capital. It is unbelievable what they are talking about in those venture meetings. And I think a lot of it is likely overstated. A lot of it is overly optimistic. Those companies will die just like our own, in round one and round two, and the equity cram downs. Those forces are at work. But if we could provide some economic reasonableness so that the responsibility of the for-profit group and the public health motivated investor includes prevention of gain of function using metrics they can use at home, then I think we have a better chance of doing that. In Russia, as soon as we talked about this version of gain of function, conversation's over. DI Ivanovsky, Institute of Influenza, or Kirk's, Budrad, Kirov, Institute of Highly Pure Preparations are no longer participating in gain of function. It's because of this language. It's just like our biodefense language. And trying to justify so much of our infrastructure for the biodefense enterprise, but that we're straying into a much more complicated discussion. So keeping the language where we keep them involved in the dialogue and giving a little bit, I think we have to be resilient in our assertions of what is gain of function and accept the fact that these developing countries need to protect their own animal stocks and their people, and need to comply with their own sovereign health security, and they can only do that with cheap, inexpensive, effective vaccines, which involve gain-of-function level principles such as massive propagation, high virulence, and conferring resistance across multiple strains. So
if I understand, I want to make sure I understood that you're saying when this comes up, they don't want to talk about it. It's not that they don't want to do the gain-of-function work, they don't want to discuss it anymore.
This is an international regulatory tax on the ability of countries to take care of their animal food populations, followed by human.
Got it. Next question.
Good morning, Gary Epstein, DHS. This panel is really focused on the benefits of gain of function, but I think it's important to let's look at what the NSAID work group has come to now. We have a lot of confusion about what the term means. There's a lot of discussion in the past about experiments that probably never should have been captured, and I'd like to have this panel crystallized on what I think we're going to be moving ahead with, which seems to be, if the NSAID recommendation is taken, a more restricted definition. And whether or not you think there should be a third criteria, the third criteria does not appear to be resistance to countermeasures. It was reframed as a different third criteria if we have one. So I'd like to start with that narrower set. But I also want to talk about some other gain of function benefits that have been talked about in the discussion previously that I don't think have been mentioned here. This is sort of the vaccine production process, most of which, as I see, is gain of function of the old type and not of the type we're now focusing on. So I think largely the discussion of the panel isn't or intersecting much with where it looks like the working group's going. But we've also heard about the importance of having surveillance and having ability to predict mutations that might confer pandemic potential, and therefore triggering surveillance systems and therefore triggering control systems. And haven't heard much of discussion about those at all, the relative importance of that type of gain of function experiment that would, the unique ability of that type of research to identify mutations, the ability of the global surveillance system to act on those mutations, the ability of control mechanisms such as poultry elimination other than vaccine production. And I'd like to know if any of you can talk about those sorts of benefits of that restricted part of gain of function that it appears that the working group is focusing on.
Oh, I was going to give it to flu, but okay. Thank you, Jerry. This has to do with just our experiences in uncommunicative countries. The requirement for them to participate in surveillance, they do at their own peril. These Ministry of Health, public health officers, and the physician officers of the foreign nations are rewarded for their own ability to produce control. This led to the fictitious reduction in Ebola rates in 2 African countries in the middle of the epidemic at a time when those rates were going up. They're being rated weekly on how many cases they have and whether or not their programs are viable. So we also know that ProMed is rife with industrial sabotage and false reporting of outbreaks that actually aren't occurring. So if anybody can put an avatar in ProMed and say, a chicken farm down the road has had an outbreak, and therefore take over that commodity market for a short period of time. So even our surveillance, and of course, recent events, a very prominent biosurveillance group is now in harm's way because of events involving their reporting mechanisms. So there's, these are casting huge prejudices and concerns about US promulgated surveillance, that it's been used allegedly for tools of economic advantage of American and other Western companies, And the second part is that under the IHR, the host country has the ability to control its own disease reporting, just as the US does. And a great example to show the inequity in the reporting is the demands that we make for MERS reporting, but our unwillingness to actually share strains for the progenitor viruses with MERS endemic countries and their labs. If you know, a great example is look how many flu strains the US government exports compared to how many they import. Okay, and to look at that, yes, we also have the CDC and the WHO reference labs and do the seed stocks and all that, but this is a continual problem that we have in justifying our demands for surveillance. So this has led to sequence in place, which we've talked about, the non-aligned nations, and also don't test, don't tell. And the example for don't test, don't tell is we know what a MERS patient looks like. Absolutely, we've seen hundreds of them, but only tens of them are being reported. And the reasons why is there's no advantage to those endemic countries with MERS to report them. They are prosperous. They are internally funded to develop their own vaccine and monoclonal strategies and to invite BARDA in to do a phase 2 study. So absolutely, they're on their way, but we cannot, I can't use the word trust. We have to understand that we've produced the wrong incentive plan for faithful, transparent reporting in the surveillance space, and that's why we have to be there. We have to breed collaborations with young investigators here and there, Europe and Africa, and because countries are more difficult to work with, those are the exact countries we got to work with. You know, Iran is a perfect one. You know, we can get DoD money into Iran. What's it take? French people. French investigators in Tehran are how we work, and we fund the French, and the French fund Iran, and then we get a Leishmania vaccine, all right, which is really important for Iran. So let's think of these new novel twists in the system that'll allow us to exaggerate our similarities, not our differences. That's my riff on that.
Very good.
Next question, please. Yeah.
Yeah, this is Gavin Huntley-Fenner. I wanted to ask a question about how this panel today intersects with the panel from yesterday, where we talked about the potential for international collaboration on standards, et cetera. And it was interesting to hear one of the speakers talk about the difference between biosafety and biosecurity, and the potential, possibly a greater potential for collaboration over biosecurity. Although today, I think we're hearing something different. I was wondering if you could talk about the ramifications of your perspective for the possibility of international collaboration.
Okay.
I think it may be up your alley again.
Okay, I apologize. So a great question, and I remember I was reflecting about great diplomatic catastrophes in the Appendix 3 review with our excellent colleagues in the FSU, where it took to day 2 in the afternoon before they realized what biosecurity meant. Biosecurity in this biosecurity meeting to them meant the protection and immune hardening of the personnel working in their BL4s, which is Russian BL3s. So wait a minute, 48 hours into an international meeting involving the G8, and we don't even have the term right. And you know, so they have a very different segmented and tiered evaluation of the elements of biosecurity. They now know the Americans mean it's locks, fences, freezers, and classified research. And they point to our NBAC and our IRF and the top security clearance of our senior scientists, and they're like, how can you tell us that you're being transparent? It is a hard thing to answer. It is a very hard question to answer. So we operate in trying to demystify it, and happily, much of the biodefense 2003 era efforts are now focused on emerging infectious disease. And so the same responsiveness, magnitude of mass casualty infections and poisonings, the same timeline issues from the influenza portfolio are being brought to bear against the Zika, the chikungunya, the dengue 2, and Asiatic forms moving through the Americas, MERS, Bundibugyo, followed by, now Meccano strain. So we have a regular opportunity to get this right. It's the beauty, the tragic beauty of emerging infectious disease, but I don't think we're capitalizing on that and getting the language out there in a way that they're really enjoying. They really have problems with American exported language that are hurting their own ability to conduct research.
Next question, please. Yeah.
Greg Koblenz from George Mason University, and just to follow up on the question from Gavin and some of the comments made by the current panelists. A lot of what we've spent today is talking about biosafety or biosecurity or dual-use research slash GOF oversight. But all of these areas are interconnected and they overlap. Is it time to break down some of the stovepipes between these categories and start thinking more holistically about something like biorisk management, which would help you integrate the things you have to do in common for all of these areas? That would maybe get around some of the terminology issues we've had and would allow you to think about how to redesign the system in order to improve biorisk management, biorisk reduction more broadly, as opposed to just tackling each of these problem areas separately.
Oh, I think that's a great idea. I think that touches on something Jonathan brought up and I mentioned as well, in that uncertainty is always a challenge, right? And coming from the FDA, one of the worst things you can do to a sponsor is introduce uncertainty. And I think, in terms of the scientific enterprise, again, if you have scientists that are on the fence about whether or not I should do this research, if you have a bar that's set, up here and it's like, well, I'd like to do it and I wouldn't mind filling out all those forms and engaging with the committees and all that, but I'm not exactly certain, how to do it. I'm not sure if I'm going to get pushback, and that's going to be a huge hurdle. So if the construct is to bring all this together to reduce the uncertainty, I don't think I have an issue with that.
Well, maybe we can wrap up with one sort of final question. I think, Jonathan, in your talk, you talked about some areas of consensus. And at least among this group of people on the stage right now, there seems to be some consensus that there's a desire not to do things that make it difficult to respond to emerging diseases, outbreaks, and produce medical countermeasures more than is necessary. Are there other areas of consensus that the panelists see that because it is getting close to policymaking, so I think it's good to not only focus on areas of disagreement, but where do you see consensus in addition to what's already been brought out?
Well, I guess I'll just say one of the points I think that I'm sort of hearing and hearing also in the questions is actually about information sharing and about making sure that the right people have the right information. And in fact, that's really, as part of the global influenza system, information sharing is really key for making good decisions about what to do, particularly in the case of what's the right countermeasure. And so I would say that sort of broadly, information sharing and assuring that actually happens is probably key. And I just sort of hear that in amongst the points that people are making.
I agree. I agree absolutely. I think that one of the things that promoted information sharing was the efforts of Flu Division, Nancy Cox in particular, who helped and made sure that the hemagglutinin sequences were available to all. So those would eventually get through the NCIB, but she felt a duty to, if she was receiving a strain of consequence from Indonesia or elsewhere, that their job was a quick turnaround and a CD, the CD would, back then, would be Federal Express back to Jakarta. So we were on the Jakarta side of that, witnessing that, and that produced a very close partnership within the CDC that was unlike any of the other partnerships with other federal agencies that were pandering for, collaborations. So structural elements are key points, and I talked about exaggerating similarities. I think that we're just, the West, including our European partners, we have a poorly pixelated views of the things that are driving gain of function. We think we know, we get committees together, but I don't see an Islamic cleric biochemist in the audience. We don't have, Persia has a really awesome homegrown manufacturing capability. Vietnam makes all of its own vaccines for childhood preventive illness, and it's Pasteur's legacy, the Pasteur Institutes. And these are groups that have played different roles over time in helping us maintain our connectivity for times of international health crisis. So, and happily, if there's a shining lining to catastrophic infectious diseases, you can be sure it's going to happen again. So each time we have one, minimal or not, is an opportunity to get it right, get a little bit farther along, go for the junior faculty, not the senior faculty, and grow your collaborations and try to make them last decades, not through 1 R01 cycle.
Well, thank you very much. We're wrapping up on time, even 1 or 2 minutes early, which is terrific, particularly since lunch is the next event. So thank you very much to everyone.
Thank you.